Hey guys, this is Caroline with Fiveable. Thanks for joining me today. Maybe some of you joined last week for the Q&A, but today is kind of open-ended, which is nice. Get some of your questions answered, hopefully, and we'll talk about the latest with College Board. I'm sure most of you have been following along. I can try my best to answer any questions you might have about that, and then we can also talk about some bio content. So lots of things that we can talk about depending on what you guys want. Okay, as always, follow us on social media. We also just got a TikTok. I don't have a TikTok, but Fiveable has a TikTok for those that do that. All right, so what are we going to be talking about today? I'm going to update you on what we know from the College Board about the AP exam. We can go over some study tips. I'll go over a little bit on how to master the FRQ, and then it's really up to you. So you guys can ask me questions. I won't be able to get into a ton of detailed information, but I can hopefully at least answer some of your questions, whether it's about the AP exams in general, or if you have some dying AP bio questions that you want to answer. Okay, so what do we know about the AP exam? So again, I'm sure most of you have been following along. Obviously, we know that the exam is going to be virtual, right? They released that, I think, a week ago, something like that. That it was going to be virtual. So everyone will be taking it at home, on the internet, or in some location, either on a computer or on a phone. As far as we know, we don't have the specific details about that. We do know that it's 45 minutes long. We do know that it's only free response. And we are 99.9% sure that it is open book, okay? That is what has been circulating around. And while College Board hasn't released that information, people within College Board have released that information, if that makes sense. So that's what we know. What that looks like for AP Bio is you're probably going to be doing some sort of experimental design question because obviously it's going to be open book. So they're not just going to ask you what something is, because anyone can just look that something up. So it's going to be all application based. So you can have the information out in front of you and hopefully it's easily accessible. So that's something that we can talk about with studying, is like finding the most important information and how to organize yourself. But then you're going to be applying that information. And then the other thing is they're going to be using all sorts of anti-plagiarism technology to ensure that your response doesn't match something on the internet or from another student. So you're not cheating. Because obviously, if they're giving it to you open book, they're not worried about you having the information, but they definitely would be worried if you had a similar response to someone else. What we do not know, we don't know how many FRQs there are going to be, right? Like the AP exam used to have 2 long and 4 short FRQs. We don't know if there's going to be 1 long, 2 longs, a long and a couple shorts. We have no idea. We will find out more information about that on April 3rd. We don't know about the timing of the questions. Are you going to be able to virtually flip through the questions, or do you have to do them all in order, right? When you have a paper exam, I always tell my students to start with a short free response. So what might that look like? What is the structure of the free response? Is it going to be, again, long, short? We don't know. And then when is this, right? They're saying that there will be 2 possible dates, but we don't know. So I don't know if it's going to be May 14th, I think is when the exam was supposed to be. Was it the 14th or 13th? Whatever that, oh, maybe it was May 11th. Wow, I'm way off. It was May 11th. We don't know if it's going to be then or if it's going to be some other time. So there are a lot of unknowns. College Board said they'll have all these answers. So Sebastian, the answer is we don't know. We don't know when the test is. It was going to be May 11th. They haven't released. They said there's going to be 2 different dates to avoid conflicts, but we don't know when those are. And unfortunately for all of us, they're not going to be posting that information until next Friday, the 3rd of April. So we have a little time to wait. So there are things that we can do to prepare ourselves for you to get prepared before we have those what we don't know questions answered. Any other questions regarding the AP exam that I may or may not be able to answer? Okay. Yeah, information is still forthcoming, but next Friday will be definitely a day where we'll get a lot more information. All right, so what can you do to prepare yourself? There are a lot of different things that you can do, but most importantly, you need to set up a good study environment. And that might be in your room. It might not be in your room. It might be like in a dining room or in a basement or, so Japesh, no, we don't know. So that was in the don't know. If I go back, the number of FRQs, we don't know. So that's the information that we will be getting on the 3rd. Okay, so setting up a good study environment. So for me, I live in a 2-bedroom apartment with my husband and my dog, right? So my study environment is a folding desk in our guest room. It is not anything fancy, but it's a spot that I can go every single day, and I know that that's my work environment, and it's not in front of the TV. It is somewhere where I can actually get focused, and you need to have a designated area for work. Otherwise, you're mixing work and play, and it can get very, very complicated. You absolutely have to create a schedule for yourself. And so there's a couple things down here that explain that. Map out what you want to get done between now and the exam. What are the units that you need to review? And I just realized that I didn't even tell you what units are on the exam. So I'll tell you that in a second. That was something that I missed from my bullet points. Thank you for the reminder, Sri Veda. But you want to map out your own plan and create a schedule for yourself, okay? So there's going to be 6 units on the exam, and we have about 6 or 7 weeks, 6 weeks. So you could do 1 unit a week. But I was talking to people, if you were joined the Q&A last week, it's good to do something like that where you can get a calendar and mark out each week a topic that you want to go over and then what you want to do each day. Maybe Monday you want to review your notes. Tuesday you want to look at study guides. Wednesday you want to practice free response questions, whatever it is. Or maybe you just want to set up a Monday, Wednesday, Friday schedule where those are the 3 days that you're going to work on AP Bio, because obviously you have other things to be worried about as well. But creating something that you can stick to and hold yourself accountable to will, 1, make you feel better, which is super important right now. And then 2, make sure that you are getting ahead of all of your goals with less anxiety, less stress, and a more structured schedule. So structure helps. Structure is very, very important. Especially for students, but for adults as well. So that's why this time right now can be really hard, because we don't have our routine. So it's important that you make a routine. So don't let yourself sleep in until 1 p.m. Because you're not going to have enough hours in the day. You have to kind of keep yourself on a regulated schedule, as hard as that is. But remember, this exam usually it's at 8 a.m. on a Monday. So if you're not used to getting up until 1 p.m. on the day of the exam, your brain's going to be fast asleep at 8 a.m. But again, we don't know the date or time of that. So I will conjecture no further. Hold yourself accountable, right? Your teachers are maybe able to, maybe not able to hold yourself accountable. Depends on what state you go to school in, okay? But you are your own best advocate, so hold yourself accountable. Use online resources, and I can go through some of these, but there's a bajillion videos, so many hours of videos that you can watch of me or someone else talking about AP biology. And then practice your writing, because what Kylie just found out is that we only have free response questions on this little 45-minute exam. So writing is going to be key, and I have some strategies there for you. So Sri Veda, to go back to, I think I actually have it here. Okay, so I have it. Maddie, we don't know the answer to that yet. So if you go back in my slides and you look at the questions that I do and do not know the answers to, unfortunately, we don't know the answer to that question. We will find out on the 3rd of April. So here are the topics that are potentially going to be tested on your free response. So it's only units 1 through 6. So units 7 and 8 will not be tested. So for your reference, what are units 7 and 8? Unit 7 is natural selection. I'm surprised actually that College Board is still having a review session on that. That's interesting. I'll have to look into that, because it's not going to be tested. Unit 7 is natural selection, and Unit 8 is ecology. So all that evolution stuff and all ecology stuff will not be on the exam. That's really weird. I'll have to look into it. Oh, okay. Brandon, great ad. He said he heard that College Board is teaching those units because colleges want students to learn it, even if it's not on the test. Right. And if you guys have already covered it, so just to put your mind at ease a little bit, if you guys have already covered, like I always teach evolution first, that's okay. Evolution gives you an understanding of everything, right? Evolution is so fundamental to biology. And the reason I teach it first is because it weaves itself into every other unit, right? Like heredity, cellular energy. There's a reason for all of these things, and it's because of natural selection. So if you learned that, don't panic, right? It can only help you understand the content a little bit better. And then ecology, it's okay. It's still really important to know. And if you guys are going to go into any science fields, it's definitely important that you know those things, which is probably why College Board is including that supplemental content, especially if you didn't get to that in class. And Maddie, I totally agree. That's why, evolution is usually the largest percentage of the AP exam. It's usually, has the largest percentage of content. But because a lot of students, most students in the country are not in school right now, they just are assuming that people were unable to get to unit 7 and 8 if they were following their pacing plan, and therefore they're not holding students accountable, just because students in all different states have different expectations on them as far as what their e-learning experience looks like. Okay, so these are the topics that you should be focused on, and just know that you don't really need to memorize a ton of vocabulary because it is open book. You're just going to need to be able to understand why things are happening. Real quick on open book. Something being open book does not necessarily mean that it is easy or easier, right? As a teacher, that's kind of like a trap because they can ask you incredibly complex questions, knowing that you have the most basic knowledge available in front of you. So with that being said, right, you might not have to spend time like memorizing things, but you also want to know that you have good resources and you know where your resources are and that you're just applying them. So the other thing is timing is going to be key, right? If you're flipping for information the whole time, you might not have time to actually write your free responses. And so it's going to be important that you organize yourself and study as if you don't have those resources so that the resources can just help if you need them. They're like a crutch, but only if you need them. I believe, Olivia, that you can use all resources, but we don't actually have that in writing yet. The only thing that we have is some people from College Board saying that it's going to be open book, but they haven't put it in their official announcement. So I'm assuming that will come on April 3rd, but I'm assuming all APs. It sounds like all APs will be open book. Maybe it won't be for bio, so I don't want to speak too soon, but that's what it sounds like, just because they're going to have a hard time regulating who does and doesn't have resources or how to prevent you guys from having resources, if that makes sense, because where there's a will, there's a way for cheating. So it sounds like this is the way that they're going to go about it, is that for all APs, they will be technically open book, but again, don't fall into the trap of that means you don't need to learn things. They could just be challenging questions where having resources might help, but it's not going to give you the answer, right? Because they still need to know who knows what, right? So I know that I don't have a lot of answers for you, and I don't know what this exam is going to look like because they just haven't told us yet. So we're going to have to kind of sit and wait until April 3rd. But what we do know we can do is focus on these units and focus on writing, because it's definitely going to be free response and it's definitely going to be these units. And those are really the only two things that I can tell you with definitiveness. That's not a word, I don't think, but now it is. Okay, so just as important as making sure that you have a good study environment and a schedule for yourself, going off of that really quick before I move on, this is invaluable for college, right? So if you're stressing, that's valid, totally valid, and I think everyone in the entire world is stressing right now. But take a deep breath and know that this is really good practice for you for college, because no one is going to check in on you in college. Your professors, you might be in a class of 700 students. You might be in a class of 100 students, right? So it's your responsibility to create a plan for yourself and map out what needs to be done and how you're going to do it. So that's your silver lining, is that this is going to make you more prepared for college. I have 2 different bachelor's degrees and a master's degree. So I've been to college more time than I have been out of college with all of that. So just know that I can tell you with definitiveness that this will help you, because having the ability to create a good routine and hold yourself accountable will make you more successful than others, which is what AP is all about, right? Okay, so 4 free responses, and we can certainly practice some of these, because then it's really up to you what you guys want to talk about for the remaining time. But some tips right off the bat that I would tell my students all the time is that the first thing that you need to do, obviously, is read carefully. There's a couple things with AP free responses that we can take a look at. They're often overly wordy, and oftentimes the words that they are using, like they might use the scientific name for an ant species. That's not important, right? So don't struggle or freak out because you see a crazy Latin name for some sort of ant species. You can just name it Ant A and Ant B or something like that in order to simplify it, because they will try to throw in heavy-duty vocabulary in order to stress you out. Also identify the action words. What are you supposed to be doing? Are you describing? Are you justifying? Are you defining? I'm so sorry. Since I'm not used to talking to other people, when I'm talking, it's making me yawn. And those are typically bolded. So in years past, the action words have always been bolded, but it's important to reflect on that action word to make sure that you are doing the action that you should be in order to get that point. And we can take a look at some free response together if you guys would like, in order to see what I mean by that. 3, outline your thoughts, whether that's physically outlining. And again, I don't know. I'm assuming they're going to allow you to have scratch paper. Who knows? But jot down your thoughts before you start writing, just so that you know you have direction. I am the worst at just starting writing and then going down a rabbit hole in which I never get to the answer. And then I end up tying up the answer with like one sentence at the end. So that is not what you want to do, obviously, especially when time is going to be of the essence in that 45-minute period. Because remember, this is a 45-minute test, not a 3-hour test. Okay. Less is more. Less is more. Less is more. I've talked to so many readers of AP exams who say that they're so sad when they read 4 pages of writing and the student only gets 2 points, right? So simplify. Don't write around something if you're not really sure. Look for what you do know and get those points, because otherwise you're going to be wasting your time. Get feedback from others. Okay. So I've been posting a bunch of discussion posts on the community board for Fiveable with practice questions, and I can write you guys some feedback. I'm sure your teachers would also do that. Maybe even a parent, a friend, a classmate, right? Because all of the College Board free responses come with rubrics, which is my second to last bullet point. It should be pretty easy for you to get some feedback from someone else. Become familiar with the College Board rubrics. So if you're not familiar with where all that stuff is, I can certainly show you, but all the past free responses are up on the internet, as well as their rubrics. Okay, so that's really, really important, because they're very specific in what they will and will not give points for. And so it's important to see what degree of specificity they are expecting of you guys. And then lastly, practice. Okay, so by the time you take the exam, if I were you, I would have done probably every free response from 2013 through 2019 and graded myself on it in order to feel comfortable and confident that I've seen enough AP style free response questions to be successful on this FRQ test. Any questions about anything that I've said so far? Take a deep breath. You're going to be okay. Hopefully you guys are spending some time for yourself and your mental health. It's very important right now. Get outside. I'm in Chicago. It's a beautiful day today. We don't have a lot of beautiful days, so enjoy your time outside. Enjoy your time FaceTiming everyone as you socially distance. But just take a deep breath. It's going to be okay. Promise. No questions? Okay. Cool. All right, so let's take a look at these topics then again. And the rest is up to you. So this is a Q&A. So you ask the questions, I give the answers. So if there's a specific topic that you want to look at, if you want to look at a free response question, if there's something that you're not sure of or you want to practice, this would be the time to ask. And then I will answer those questions. But since it's not one of our structured like Wednesday night or Monday night cram sessions or anything, macromolecules, FRQ, G protein linked receptors, yes, Unit 1 FRQ, Sample question, signal transduction. Holy moly, so many different things. Okay, perfect. Yes, we can talk about all of these things. And actually, a lot of these are related too. So we can certainly take a peek. Okay, so I'm hearing a lot with this biochemistry stuff. So macromolecules, G protein linked receptors, and signal transduction. We can combine all of those, and we can look for an FRQ of those as well. Okay, give me 1 second, just because I obviously have to do this live. All right, so for signal transduction, we're going to bop over. If you haven't done any work all year, I'm going to say you have to work really hard the next 6 weeks, Sebastian. Okay, so question 8 of the 2013 free response is going to talk about signal transduction. Okay, and then I'm going to show us another one with a more complicated signal transduction. But take a look at this one right now. And you can kind of be asking yourself what you would include in each section, and then I'll tell you what I would include. Olivia, the FRQs changed after 2013. 2013 was the rewrite. So 2013 on you can use. Okay, cool. Sounds like people are ready for me to go over it, so I shall. So I'm actually going to pull up for us the rubric to this, because you should become very familiar. So I just posted a link before I go over this for Callie, which I'm really glad you asked, that has all of the past. Usually I just Google like AP Biology free response 2013 and it pops up. But it has all of the past years of free responses, as well as their scoring guidelines, which is what I'm about to show you guys, which is the rubric. But it also tells you the score statistics, and it shows you sample free responses. So I'm going to show you what those resources look like once we go through the rubric for this. But it's really good to see what a sample student response looks like, so you can see what really good writing is and what really not great writing is. And then you can see where you fall on that spectrum. So here's the rubric for this question, because I want to make sure you guys are being specific enough, because I cannot tell you how many times I have not been able to grant points to my students because they're not specific enough. All right, so here's what you needed to include. And from what I read in the comments, it seems like you guys did a really great job, but here's where you need to be careful. So step 1, so we needed to explain the role of each numbered step in regulating target gene expression. So explain means you need to expand upon. You're not just defining, you're not just writing what the thing is, you're actually explaining how it contributes to regulating target gene expression. So explain is a hard one. You need to be writing a decent amount for that. So step 1, and a lot of people said this, we either have a hormone or we have a ligand, so they don't care what word you use, binding to the receptor. A lot of people would be able to write that. That's not enough to get you a point, because that's what's shown in the diagram, right? I could pull any student and they should be able to tell me that just by looking at this. As an AP student, and here's where this is an application question, it'd be very hard to cheat on something like this. You need to be able to say that that initiates, triggers, or induces, or starts any of those action words, signaling or signal reception. Okay, so it's not enough to just say that the ligand binds to the receptor. Even though you're using the appropriate vocabulary, you have to say what that means as far as how that's regulating gene expression. How is it regulating gene expression? Well, it's initiating the signal response. Okay, hopefully that makes sense. So again, you're not just naming the thing. Okay, step 2, so we have an intracellular cascade, okay, or we have, I saw someone say a transduction pathway. That's perfect. That either transduces, amplifies, or transfers the signal from the plasma membrane to the nucleus. Again, you need to be that specific. You can't just say it's a signal transduction pathway, okay? You have to say that it is a pathway that transduces the signal from blank to blank, okay? Because that is its role in regulating target gene expression. Does that make sense? So this is where students oftentimes lose a lot of points, because they know that, but they're just not specific enough when they're actually writing it down, okay? So you have to put whatever's in your brain on the paper. And then step 3, hopefully you said transcription or gene expression of the gene is activated or repressed, actually. So you could say either, right? Because a lot of times we're actually repressing a gene or turning it off. So saying, so Angela, I'm not picking on you at all because this is something that I would say or my students would say all the time, saying the responses produced, you would need to be a little bit more specific than that by saying how. So we have transcription occurring, or we're expressing that gene is being stimulated, that gene is being repressed, right? And that's how that's actually contributing to gene expression. So again, it's not enough just to name what's happening in each step. You need to understand how it's contributing to gene expression. Explain and describe. So describe, you're going to give more details, and explain is more part of a process, but that's kind of specific. For the most part, describe and explain will be pretty similar. Define would be different. Define, you can just give usually like a one-word answer or a very short answer to that, as opposed to explaining or describing where you have to elaborate more. Okay. Awesome. So that was an easier free response. So just for reference, I want to share with you really quick, because this is a good resource, because the best thing that I can do is prepare you to do this on your own, because that's how you're going to be learning. So here I'm under 2013 free response questions. Here are the actual questions. Here are the scoring guidelines. All right, here are your scoring statistics. Okay, so what you'll notice is that the average score for question 8 is 0.9 points, so less than 1 point. Okay, so each one of those parts, explaining step 1, step 2, and step 3, each one of those was 1 point, and the average was less than 1 point. So if you got 1 out of 3 on this, you're in good shape to get a 3. Okay. So sometimes it's important to see those statistics. So you can also see, like for some of those longer questions that were worth 10 points, they have really low averages. You don't need to get all of the points to get a 5 or a 4 or a 3. You just need to get more than other people. So sometimes I look at these statistics all the time, but if you're struggling with a question and you get back like 4 points out of 10, you're actually in good shape to get a 3 or 4 with that. So try not to be so hard on yourself. And then there's also, if you go back and look at that page, I'm not going to do it right now just because everything is taking so long. You can take a look at other student responses and how they did. And you can even practice grading them to get a good idea of what is a good and what is a not so good response. Okay, so I find that very helpful. Hopefully you guys do as well, just to see what other students like you are writing and if you need to step up your game or if you're doing well. Callie, what year would be the best to look at? I would look at all of them because they're all going to have different information, but obviously know that evolution will not be and ecology will not be on your test. Okay, so I wanted to show you another example, just how they can combine a lot of different materials. This is a very challenging question. This was one of the long free responses from 2018, so 2 years ago. And I can give you guys the link to this as well. It's going to be question 2. And then I know people wanted to talk genetics and chi-squared and all sorts of stuff, so we can try to do all of that too, and macromolecules. Obviously, you can see we have a pathway here that's also involving a lot of different molecules. And then this question is also about cellular respiration, which is just crazy. So they're combining a lot of different things here. But let's just take a look at A, B, just A and B, because A and B, the rest are going to be related to a lot of different units. So that's something else that you need to expect, is that all of these questions are going to involve probably multiple units from units 1 through 6, right? Like this one involves a lot of unit 4, 3, 2, a lot. Probably 4 or 5 different units would be covered by this question. But take a look at A and B. Okay, and hopefully you can pull this up on your own computer so that you can look at the question or the diagram as well. And then we'll talk through these answers. Something important to remember is that you're not expected to know what these things are. Okay, so don't get caught up and think that you don't know anything. You know lots of things. Okay, hopefully that was enough time. Someone yell at me if it wasn't. But I want to go through the answers to these first 2 just to show you that there are going to be easy points that you can find. Okay, and this is something that I stress with my own students all the time, is find the easy points. Okay, so here we go. These are easy points, guys. All right, So how can we first figure out how many points this is worth? So describe, that's bolded. And again, that's very similar to explain. We're describing two things. There's this giant and here. So that's going to be worth 2 points. Okay. And then we're making a claim with a reason. Okay. So claim, reason, another 2 points. So part A was super simple. Describe the effect of inhibiting step 3 on the formation of pores and on the release of interleukin. Okay. So I'm going to go back up and look at this. I'm going to find step 3. How is this going to affect interleukin? And how is this going to affect our pores? Okay. If we inhibit step 3, pores will not form, right? Let's take a look. Step 3 leads to pore formation. That's a pore. If we inhibit this, then we're not going to be able to get our pore formation to happen. Is inhibiting step 3 going to do anything to interleukin over here? Is interleukin depending on that pore? No, right? It's going right through the membrane. It's not using this pore. So interleukin will be unaffected, and that's 2 points just from looking at the diagram. A point for saying pores will not form, a point for saying interleukin release will not be affected, or interleukin release continues, or interleukin will still be released, anything like that. So those are 2 easy points. All right, let me look back. Someone had a question. If the interleukin is released, sorry, you're answering the next question. So hopefully that gives you some peace. Now let's look at part B. Make a claim about how cleaving inactive caspase 1 results in activation of caspase 1. A student claims that pre-infection production of inactive precursors shortens the response time of a cell to a bacterial infection. Provide one reason to support the student's claim. Okay, so first we have to make our own claim about how cleaving inactive caspase results in active caspase. This is, again, super easy. So this goes into macromolecules. So whoever was asking about macromolecules, proteins are going to be probably the most tested, that and nucleic acids. So those should be your focus. But all you had to say is that this removes an inhibitor, this removes a repressor, this removes something, or it changes the shape of the protein, right? Do you know that to be true? No, we don't know how this thing works, but you have a good science brain. So trust your science brain and know that this is how proteins usually work. This is how enzymes usually work. We have an inhibitor, or we have something that we're removing, or we're changing a shape that changes the activity level of that thing. Okay, so you can actually think really broadly there. And when all else fails, go back to your basic biology knowledge, right? Because that's basic biology knowledge that you just overcomplicate in your brain. Okay, and then we have a student that says pre-infection production of inactive precursors shortens the response time, so we have to support their claim. So you could have said that cleaving is faster than making one upon infection. So if we just have to cut off the repressor as opposed to making the entire protein, that's going to be faster. Or we don't have to wait for transcription and translation or protein synthesis, right? Because the protein's already made, we just have to activate it. So saying anything like that would get you your 4th point. So for reference, the average score of this question in 2018, I pulled it up, was 2.68. No, 2.48. Let me double check. 2.68. Okay, so less than 3 points. So if you could get those 4 simple points in the beginning without knowing a whole ton of information, you're in pretty good shape to pass this exam, right? Then it gets a little more complicated. And I'm not going to go into parts C, D, and E right now, but you should certainly take a look at them. They're not quite as complicated as they look, but students were terribly scared of this question. So it had very, very low scores. One of the lowest scoring, because remember, this is out of 10. So if you get a 2.68, you're getting 26% of the 27% of the question right. Okay. Okay, so other things that people wanted to go over. Oh, G protein linked receptors. So, okay, so I can use this question as an example of that, Angela. G protein linked receptors just mean that they need GTP in order to be activated. So GTP is just another version of ATP. They're very, very similar, and they just need that energy in order to be active. They need a GTP bound to them. So it's kind of like how we see here, we have some inactive and then active things, although with all of these, we're removing a repressor, so it's not exactly the same. But we basically just have inactive G proteins that just need a GTP, which again is very similar to ATP, to bind to them in order to be activated. I would not expect that you would need to know that detail of information. But you can tune into the unit 4. Yeah. 3, 4, 2. One of those unit reviews. I'm forgetting where it is. And I'll definitely talk more about all the different types of receptors on cells. Should be unit 4. That's cell communication. I was blanking for a second. Okay. That's that. We've talked about signal transduction. We've talked a bit about macromolecules. That's what I'm going to be hitting really hard on Monday, but you need to be familiar with like what atoms make them all up. So everything has CH and O. Then we have amino acids, also have N, P, and sometimes other things. And then nucleic acids have N and P as well. Amino acids don't always have P. They can have a lot of different things, but they definitely have N because they all have amino groups. But then we always have nitrogen because we have nitrogenous bases, and we always have phosphorus because we have the sugar phosphate backbone in DNA. So you should know things like that. You should know that proteins denature at various pHs and heats. Right? You should know that enzymes are proteins. You should be very familiar with enzymes and their structure. If they get too hot or if they get too cold or if they're out of their pH range, they will denature and stop functioning. Yes. All right. Cool. Yeah, it's only different because it needs GTP, not ATP, Angela. So hopefully that answered your question. Sriveda, I know you asked about macromolecules. Let me know if there's anything more specific that I can go over. I see some heredity, help with the process of DNA replication. All right, so let's see. We have 15 minutes left, so I'm going to do a poll. Ooh. Derek, how important are functional groups? Not important at all. You mean functional groups of amino acids? Not important at all. Don't worry about them. Definitely don't worry about them. All right, chi-square. Unsurprisingly, chi-square. All right, so we're going to take a look. Y'all, if you were with me last time, one of my predictions is that chi-square may be very important on this exam because if you don't know how to do it, you can't briefly learn it. So it's a good way to test your knowledge, and it hasn't been covered in a while. Okay. William, you're good. Yeah, if you know what an amino group is and a carboxyl group, you're more than fine. All right, so we're going to look at a free response of chi-square since we know that free response are the name of the game for this AP exam. So we're going to look at the 2013 again. Wow, 2013 had a rough year. Okay, and this is going to be a free response. They haven't done one with heredity. So when we cover the Unit 5 heredity review for the cram session, which again, I'm going to post the link of how you guys can get a cram pass. I'm going to go over, I'm going to for sure go over a chi-squared with heredity because that very well may be on the exam. So I'll make one up before the cram session on heredity, which will be, when is the Unit 5 session? I can tell you guys. That will be on April 27th. That's when the Unit 5 review will be. So pretty close to the AP exam as of now, unless things change. And again, we need to hear from College Board about your exam date. But I'll definitely go over one of those, which will be perfect. But for now, we'll take a look at this animal behavior chi-squared, and we're not going to go into all the parts. We'll just go into the ones that are related to chi-square. Okay, so in an investigation of fruit fly behavior, a covered choice chamber is used to test whether the spatial distribution of flies is affected by the presence of a substance placed at one end of the chamber. So choice chambers are very common, and I've seen multiple free response with chi-square related to choice chambers. Okay, so they have a glucose-soaked cotton ball on one side and a dry cotton ball on the other. To test the fly's preference for glucose, 60 flies were introduced into the middle of the choice chamber at the insertion point indicated by the arrow in the figure above. A cotton ball soaked with 10% glucose is placed at the other end, and a dry cotton ball is placed at the other end. The positions of flies are observed and recorded every minute for 10 minutes. Okay. So the first thing that we need to do is predict the distribution in the chamber after 10 minutes and then justify our prediction. So predicting is super easy. We can say one word. Where do we think these flies are going to go? Where are the flies going to go, y'all? Yeah, to glucose. Perfect. Y'all just got a point. Good job. They're going to go to the glucose-soaked cotton ball. But why? So the why is the justified part. Yes, flies like sugar, William, but why? Why do they like food? What are they using for a source of nutrition? Be specific. Good. Glucose is needed for energy, or sugar is needed for cellular respiration, or sugar is used to make ATP. That is perfect, y'all. So that is how you get your justified point. Okay. So instead of just saying it's a food source, go a step further. What are they going to use it for? They're going to use it to create energy. They're going to use it to create ATP. They're going to use it in cellular respiration. You guys know that. I know all of you know that, right? So just make sure it ends up on your paper, because unfortunately the teachers grading your free response don't know that you know that. They assume you do, but they don't know for sure. Okay. Wow. Everything has to somehow relate to biology in the end. Exactly. Yes. And that's why if you went over evolution, you're still going to be a total killer at this exam, because all of this relates back, right? It makes sense. They're going towards that end because it's evolutionarily beneficial to them to make energy. Okay. So the next part is to propose one specific improvement and explain. So an improvement to the control and an improvement to the environment. So we're not going to go through that part just because it's not directly related to chi-square, but that's something to think about. I'm guessing, right? That's not something that you can cheat on. You can't cheat and find out what a better experimental control would be, right? You have to be looking at this specific question to be like, oh, maybe the cotton ball should be wet, because how do we know that they aren't going to the water that's in the glucose-soaked cotton ball and the glucose, right? We don't know. We do know, but it would be better to test it with a water-soaked cotton ball. But those kinds of things you can't cheat on. So a wider insertion point perhaps, or maybe we want to make that middle region a little bit bigger if we're going to count it. I forget what other environmental factors they list. Let's see what else they list. Brief pause. I said we weren't talking about that section, and then we started talking about it because you know what? It's actually really good to know. So they also said you could use different concentrations of glucose, different temperatures, different light levels, different choice chambers. So you could change the size, so your answer would count. Vary the duration, vary the time of day, and all of those things, right, are to ensure that glucose is the only thing that is affecting the fly's choice. Yes, light, temperature, good. All of those things are great, guys. You're thinking the right way. So we could have several cotton balls. That's a perfect explanation. So all of those are really great, and it shows me that you have a strong science brain and you're thinking through this very strongly. So that's your proposition. So that's your proposal. We want to wet the cotton ball. We want to add light. We want to make sure there's no other smells, whatever it is. You get 2 points for your proposal. The other 2 points come from explaining how the modification will affect the experiment. And that's where you need to talk about whatever you're putting into place should enhance the scientist's ability to ensure that the flies are choosing the glucose and nothing else, right? That's why we have controls. That's why we control the environment. That's why we have experimental controls, because we want to make sure that the flies are merely responding to the glucose and not the sun and not another smell and not the water, right? We want to make sure that they're just responding to the glucose. If the grader agrees with you, but it isn't on the answer sheet, is it correct, Sarthik? That's a great question. For something like this, most likely they would grade it correctly just because there are so many possible responses. So usually on our rubrics, it would say something like, or something similar, right? So on this one, it says includes but not limited to, and they give a list, right? But if you still say something that would be right, even though it's not on the sheet, then that teacher would still be able to give you points. Okay, so that's one of those, which is a really good thing, because it's not frequent that we see something like that. So I would become really familiar with experimental design. Why do we have controls? Why do we have environmental factors? In order to ensure that our independent variable is the only thing affecting the variables. That should be ingrained in your brain. You should be ready to write it down, because I would bet that an experimental design question is coming your way. Okay, we haven't even started talking about chi-square yet. Here we go. Alrighty, let me make this a little bigger. The experiment described above is repeated with ripe bananas at one end and unripe bananas at the other end. Once again, the positions of the flies are observed and recorded every minute for 10 minutes. The positions after 1 minute and after 10 minutes are shown in the table below. Okay. So we need to perform a chi-square test on the data for the 10-minute time point, okay? So 10-minute time point means I'm just looking at this row right here. This is the data that I'm using. And then I need to specify a null hypothesis and enter the values from the calculation in the table. So that's 3 points right there. And then we get more points for justifying our hypothesis. Okay. So the first thing that I would do is in this observed section, that is where I'm going to fill in 45, 3, and 12. We observe those things with our eyes, right? That's actually what we saw. And that adds up to 60, which is perfect because we know we have 60 flies. And so we need to have that as our total. Any ideas what our expected values would be here?
What do we expect? Maddie, you crushed it. 20. Yes. Good. So
sometimes students get confused. Uh-oh, can you guys not hear me? Is it better now? Is it still bad? Okay, but going back to Maddie, expected is going to be 20. Chi-squared is evil, William. That's probably why. 20 throughout, 20, 20, 20. How do we know that we're right? Because our total is also 60, okay? Students get this confused all the time as well, or a lot of my students want to put like 30 and 30 for both ends and none in the middle, but we're expecting no influences here. So we're expecting an even distribution. Just for these choice chambers, your expected values won't always be equal, right? If you're doing a chi-square for heredity and you expect 75% of the offspring to look one way, then you should expect, have that in your expected values represented. Okay. So then how do we do the actual math? So I don't have the capability to write this out for you guys right now, but I will when we do this in the heredity unit review. But we know, and this is always given to you, you don't need to have it memorized, that we always do observed minus expected squared, those parentheses matter. And then you're going to divide that whole thing by expected. So we can do one together now. Let's just do our observed and expected for the end with the ripe banana. So our observed was 45 and we expected 20. So we'll just do that. I'll give you a second to get those answers. So Sriveda, the expected in this case, we are saying, and I should have mentioned what our null hypothesis would be. Our null hypothesis would be that the bananas have no influence on the fly's behavior. Okay, so our expected values are going along with that. We're trying to disprove that hypothesis and say that the bananas actually do affect the fly's behavior. Okay, and so that's what we're doing the chi-squared to test. So chi-square always tells us if our results are significant or not. So our null is always saying that everything is due to chance and that our independent variable has no effect on the results. So again, the bananas would have no effect on the results. So due to chance, we would expect an even distribution of 20, 20, and 20. You would also be given the critical number, Maddie. Yes, and I am thinking that you have a good understanding of chi-squared. Yeah, so our null hypothesis would be the number of flies in each section stays the same. They would be evenly distributed throughout. Yep, absolutely. Okay, so for that chi-squared value, you should have gotten 31.25, and that's by doing 45 minus 25, then squaring that answer, so 25 squared, which gives you 625, divided by our expected, which was 20, you should get 31.25. Okay, and this math actually, I'm sure if I look at the answer key here, they give you all the math. Where did that go? Why do I keep losing things? Okay, I'm going to post these answers so that you guys should be looking at these instead. But I wanted you guys to see. And then we'll call it quits here in a second, but we'll finish up this question. So Tim, in this, no, it's not a strange question because especially since you're given that like 21, 18, 20 or whatever it is as their starting position, their position at minute 1. Oh no, I wonder why. Sorry, I probably just blew someone's eardrum out. Is it better now? Is it still loud? It's better. Oh, maybe that is it. Okay. It doesn't like when I switch the screen, maybe. Yikes. Sorry, guys. Sorry for whoever said they had AirPods in too. Oops. We're expecting nothing to have an effect on anything. So Tim, you're exactly correct. It should be 20, 20, 20 because nothing is having an impact here, okay, according to our null hypothesis. So we can see all of this here. The null is that the flies will be evenly distributed across the 3 different parts of the choice chamber, or you could say that the null is that the bananas will have no effect on the fly's behavior, anything like that, just says that there's no effect on our outcome. Then obviously you can see the calculations there, so you can check yourself, and that total value is massive, okay? So you get a point for your null, and you get a point for your expected value, and then the rest of your points come from explaining if, yes, the expected is the null. That's a great way to say it, Eve. Thank you for summarizing that so beautifully. Okay, so then we have to take a look at our degrees of freedom and our p-values. Okay, so you're always going to use the critical value for p of 0.05, and that critical value for this is 5.99. So you would be given the p-value chart, and I can go through that with you guys when we have more time. But degrees of freedom is always the number of possible outcomes minus 1. So our possible outcomes here were end with ripe banana, middle, and end with unripe banana. So we have 2 degrees of freedom. And using that chart, the p-value chart, which you will be given for degrees of freedom of 2 and a p-value of 0.05, the critical value is 5.99, okay? You do not need to use the p-value of 0.01. It's too specific, okay? So knowing that, our number is way more massive than the critical value. 48.9 is way larger than 5.99. And so therefore our hypothesis should be rejected, okay? Think about it. What we just did was find the difference between what we observed and what we expected, and the difference was massive. What that means is this is not right. The flies are not evenly distributed. They're all over the place, mainly towards the ripe banana, which makes sense because bananas have hormones that they emit, right? So when this number is really, really large, we reject the null. This is not due to chance, right? Our differences are too big. That's why this number is so large. Mathematically speaking, that should make sense that we will reject our null hypothesis, and you needed to explain why. So you explain that your hypothesis is rejected, and then you justify it by using the degrees of freedom and the p-values, okay? Yes, that's why the degree of freedom is 2, because you have 3 possible choices minus 1. Do you have a math section? So Kylie, if you had any math, it would be embedded in a free response. It's only a free response test. The old test for you guys also, even the multiple choice for you guys wasn't going to have the typical math section that it used to have. But there could be math, like this was a free response from 2013. So there's obviously math embedded here, but in its own special way. So I can go into more details as far as like the chart, degrees of freedom, p-values, etc. I also did a stream a long time ago that I'll try to find to you guys. And when I find it, I'll email it out to everyone who attended today, of myself going through this in more detail. So that will be helpful just because we're out of time for today. But hopefully that's helpful to you guys. Okay, so to wrap it up, Q&As, keep coming with your questions. Cram Pass, get your Cram Pass. I'll be streaming a unit review and so much free response practice. You guys have no idea. So come with your questions. That's what I'm here for. You guys are very, very capable. Go back and rewatch and listen to the things that you should be doing to set yourself up for success, and know that you will be okay and that we will all grow from this experience. And it does have an end, and you will be stronger on the other side. Yes, those videos of the reviews you can only get with a Cram Pass. Other videos are free and you don't need a Cram Pass to rewatch. Okay, so that video of the chi-square I can send to you. That's free. But if you want to come to my cram sessions, then you need all that. Okay. Bye, guys. Thanks so much for tuning in. I really appreciate it. Hopefully I see you soon. If not tonight, I'm talking about cellular respiration and photosynthesis. See ya.